candy coloured slides are not going to project desperately well, but we'll see how we go. So I'm Aimee, so I'm local to Manchester, and so it's really lovely that you all came to me rather than me coming to you guys. And hopefully, what have we learnt so far from doing gene therapy trials in children with, let's say, brain disorders, but neurodegenerative diseases is what I'm familiar with? So what I would say is that it is possible to treat inherited disorders, so genetic problems which affect the brain and development with gene therapy. So we've learnt that so far. We need to figure out what the best way to deliver that therapy is. We need a really good product. So when I say product, what I mean is the gene therapy that we give needs to be good. It needs to do what we want it to do. Sometimes we reject therapy, so gene therapy can be rejected by our bodies, and that does happen. And the way that we design our clinical trials is really, really important to the success. So these are some of the key things that we've learned. And interestingly, I put this slide in before I'd seen Dan's slides this morning. And I used this slide because this was a little bit more about my journey with rare disease and gene therapy. And I think of every bend in the road has been my meeting of another patient, dosing of another patient, because every time I meet a patient and I work with them in a clinical trial, it changes what I do for the next patient. So I learn at every point of the way, and it's really tricky and it's a hard journey, which you all know because you're on it. And this is going to be a bit of Groundhog Day. Not everybody in this room is going to get this reference, are they? But because we've already heard most of what I'm going to say today, but I'm going to go over it again, so I apologise for the repetition, but this is where we're at. And some of my talk might feel a little bit jaded and a little bit pessimistic, but I promise you I'm a glass half full person. So I am at heart an optimist, but I'm going to say some things that are going to be difficult to hear and things that are going to make you feel like I don't believe in this. And I'm going to try and convince you that I do believe in gene therapy, but I just want you to be realistic about it. So we're going to get to the smiley face hopefully by the end, but it might be a bit dodgy in the middle. Okay, so we've seen in the news recently all of these fabulous gene therapy trials, and so you hear all the time at the moment about a new gene therapy has been approved, and so that gives us all loads of hope. But what I want us to understand a little bit more is about what these different types of gene therapy are and how it relates to gene therapy for Rett's. So I work in a group of disorders called lysosomal disorders, and essentially you have some stuff which is converted by an enzyme, which is a protein, into some other stuff, which might then be converted into another protein or product. So you have this process going on in your body and your cells all the time, and these enzymes are encoded by genes. And so you might have a whole process. If you haven't got gene X, it means that you're not going to make enough of or working enzyme 1, which means that stuff at the beginning can't be converted to your product or substrate B. And this becomes a problem for us because that buildup of stuff at the beginning, your substrate, could be toxic to your cells, so it might do damage to your body. The whole process might be essential for making energy, for doing something really important in your body, or the final product, which you're going to have less of, might be absolutely essential for your brain function. So this is the kind of problems that I deal with primarily. So it's a little bit different to Rett's, but it's the same kind of idea. And in my storage disorders that I look after, we have this problem happening in lots of our body tissues. So it's happening in our lungs and our liver and our bone marrow, so it's all over. But it's also happening in the brain. And that's why we're now thinking about gene therapies for these disorders, but we need to get into the whole body. So it's a little bit different to Rett's. And in neurodegenerative disorders, we've already heard about the difference with Rett's, but just so that you understand the position I'm coming from, these are disorders where I might have a baby or an infant who's developing perfectly normally, is doing really well, and at some point they start to lose skills and they continue to lose skills. So this isn't the same as Rett syndrome, but these are the babies and the children that I'm looking after. This can happen in the first few months of life. They may lose the ability to feed, the ability to swallow, the ability to walk, communicate, and they'll reach end of life without any intervention. And for most of these disorders, I have absolutely nothing to give them. I have no therapy. And I've got a window by which I want to try and intervene before it's too late. As you all know, this is not the same with Rett syndrome. And as we've already heard, we're not losing brain cells, and your children are still gaining skills, which is really quite a different scenario. So today, I'm feeling a lot more positive about working in Rett syndrome than the conditions that I currently look after. So when I'm thinking of treating lysosomal neurodegenerative disorders, I'm trying to stop that disease progression. I'm never going to reverse what's already happened to the children that I've got, which we might be able to reverse with Rett syndrome. The rate of progression of the disorders I'm looking after is so fast that I'm usually too late. We're not seeing the same with Rett syndrome, so we're in a different position. We still have the same challenges that we still need to get into the brain. I need to get it to the key parts of the brain, so it's not that I just need to get it in there, I need to get it to where it needs to work. And I need to keep that gene therapy continually active, and which is a big challenge we've not talked about today. And I need to stop the body from rejecting both the thing that's carrying the gene therapy, which we call the vector. So you've heard us talk about AAVs, and we've mentioned vectors today. So that's the kind of transport mechanism by which we give the healthy copy of the gene we're trying to get in there. But also the thing that the gene is making, so the protein. So you might reject the vector, but you could also reject the gene product. And we've touched on this as well. When we're thinking about ultra-rare disease or rare diseases, there's a spectrum of the phenotypes, so different patients look different to one another. So that's a challenge. And you've heard about these challenges already today. So this was Dan's slide from this morning. And so you'll hear that I'm saying a lot of the same things that he told you already. So if I'm designing a gene therapy trial in a neurodegenerative disorder, I need a really good therapy, I need a good product that's going to do what I want it to do. We need to think about how we're going to get that gene in there, and I'm going to talk to you about ex vivo versus in vivo therapies. We have to pick what our transport is, so which vector are we going to use to get it in there, and why might we use a different one. We might attach something to that gene therapy or to that vector to make it go to the cells we want to get to, so we can pick different what we call promoters to make it go to those places. We need to pick how we're going to give it, which is the best injection route to give it. What's the best immunosuppression regime? Who are the best patients for that particular therapy and that particular trial? Are we going to increase the dose? How are we going to pick the dose? And how are we going to know whether it works? So we have to think about outcome measures. And we have to have our long-term plan for how we're going to get this in the current economic climate to you guys, to our patients. So we're going to talk a little bit about types of gene therapy, and I just want to touch on this just so that you have a bit of an understanding that when you hear certain things about gene therapy in the news, you kind of got an idea that there's different types of gene therapy out there. So we've got something called in vivo and we've got something called ex vivo. So in vivo are when we inject, the way we're talking about today, so the TACER and the neurogene products, we're going to inject that gene therapy directly into a body compartment. Ex vivo is when we're going to take somebody's blood cells out of their body, give their blood cells the gene therapy, and then give those cells back to the patient. So it's a little bit like a bone marrow transplant. And I'm going to tell you that because this news article about Teddy and Nala, who have metachromatic leukodystrophy, and was the most recent kind of big news story about gene therapy, that's an ex vivo approach. So that's something really different to what we're talking about for Rett syndrome. Spinal muscular atrophy, which you'll have also heard of, which has a gene therapy, is the in vivo approach. So that's more like what we're talking about for Rett syndrome. And so there's big differences. So when we talk about ex vivo therapy, we're basically giving a gene therapy everywhere that the blood goes, the gene therapy should go. We use what we call integrating gene therapies here. So we're using vectors which integrate into the patient's genome. So once it's in there, it stays there forever. And there are risks with that approach, and there's kind of cancer risks are the thing that people worry about most with that. And in this type of gene therapy, we use a modified HIV vector, so a lentiviral vector. So it's a different type of vehicle to take that gene therapy around the body. And this is a slower approach. So we have to take a patient's cells out of their body. We then have to send those cells to wherever's going to manufacture and implant the gene therapy. And then they have to go through a whole bunch of quality control mechanisms. So it's a few months down the line before we can give that gene therapy back. So for some of my conditions, I don't have 3 months to wait for my kids. So it's not really appropriate for every patient and every disease. But we still need that gene therapy to work and to do what we want it to do. In vivo gene therapies, you're targeting where you want that gene therapy to go the most. So sometimes we use in vivo for a liver disorder, and so we want it to go directly to the liver. Sometimes it's for a brain disorder, and so it's more specific to a specific tissue. And these AAV-based gene therapies are what we regard as non-integrating. So they won't necessarily stay producing that product indefinitely. And we don't know exactly the biology of that at this point in time, but it's not quite the same as the lentiviral products. And there's probably a benefit in terms of risk of where that would integrate if it were integrating. These are faster, because you give it as soon as you've gone through your process. So you've recruited to a trial or you've got an approved therapy, you just dose. So it's an injection of something somewhere. But again, we still need it to be a good product. It still needs to do what we want it to do. So we talked about this morning, about where do we want to inject the gene therapy in a neurological disorder. So we can do intraparenchymal injections, so right into the brain tissue. But that's no good if it's your whole brain that you want to treat, because otherwise you'd have to put an injection in all over your head. It's not going to work. It's not a feasible solution. It works if you've got a specific part of the brain that you know that you need that gene therapy in. So that's quite unique. We can do intracerebral ventricular, so into the fluid-filled spaces in the brain. We can do intracisternal magna, so this is the base of the head, so it's an accessible, I don't want to say easy, but it's an accessible point to get into the CSF. Or we can do intrathecal, usually in the lumbar spine, so we go in like a lumbar puncture and inject there. We could also give some gene therapies IV, so into the bloodstream, and then tell them to go to the brain. That's probably been less successful so far, but it's not an impossibility in the future. So we mentioned immunosuppression. So there's 2 reasons why we need to immunosuppress in a gene therapy trial. One is if we don't want the body to reject the vehicle carrying it, so the virus. So they're kind of deactivated viruses. So we're kind of taking advantage of the fact that viruses get into cells. But we don't want to cause infection, and your body might have seen some of these viruses before, and so we don't want your body to reject them when they're carrying something really precious like a gene therapy. But also for some of the gene therapies, you're giving a gene to a patient to make a protein that body has never seen before. And so there's a chance your body will go, well, I've never seen this before, it must be foreign, we need to reject it. And if it rejects it, then even though the gene therapy might have worked, it's not going to work anymore because your body's fighting it off. So we have to think about how are we going to stop your body from rejecting it in the safest possible way. And that's really tricky. We don't really know what the right thing to do is, and so every clinical trial seems to have a modification on a different immunosuppression regime. And it's really important that we think about that because we see different reactions in different patients, and often the reactions are to the immunosuppression regime that we're giving them, but we know it's so important that we don't reject this gene therapy that you've had a once-in-a-lifetime shot of getting. So We do lots of additional tests to monitor for those reactions and to also monitor for responses to the immunosuppression. So this just means that when you're taking part in gene therapy, we're going to do a lot of extra safety testing that your child would never normally need in any other circumstance. And these medicines, so they might be steroid medicines, which all come with side effects. They might be drug infusions that you might have to have on a regular basis, so biological therapies. And they might be things like anti-rejection medicines. So these are things that we would give if you were having a kidney transplant and we're wanting you to stop you from rejecting that kidney transplant. And so these are drugs that need regular monitoring, but they also have side effects, not just infection risks, but you might get hematological changes, you might get rashes, it might make you sick. So there's lots of side effects to all of these medicines that we're giving you just so that you can hold on to that gene therapy that we've given. The thing with all of the immunosuppression, as with all of this, is that every child, even if they've got the same disorder, they're still different. So, you know, if I take paracetamol, I might be absolutely fine, but my partner might not be. He might have a reaction to paracetamol, and there's nothing I could have done to predict that. So we just have to be mindful that just because 5 out of 6 children in the clinical trial didn't react to the immunosuppression, the 6th one didn't. If that 6th one is your child, that's really important. So I'm going to try and take you on a bit of a kind of very practical tour of what it's like to be part of a clinical trial. This is our clinical trials unit in Manchester, and my boss Simon there will be very pleased that I've put him on a big picture for you all to see. So how do you, and this kind of came up this morning, how do you get into a clinical trial? How do you access a clinical trial? So like I said this morning, I'll take a self-referral. It can be your doctor. I've had family members email. So there's usually, for all clinical trials, and you're really fortunate because you're all part of Reverse Rett, so you'll be told as soon as the clinical trial site opens. And usually you'll be told what kind of patients roughly would be included in that. And you can usually just email us and we'll tell you whether we think you might be a candidate or not. We then, as a group delivering a trial, will sort of pre-screen patients. So we'll kind of look at broad characteristics. How old are they? Do we know if we've got their genetic test results already? Do we think that there's a chance that they might be eligible? Because I don't want to bring you to Manchester for me to say, oh, actually, no, your child's 16 and I'm only recruiting 2-year-olds. It's just a waste of everybody's time and it's a great disappointment. If I see that somebody actually might meet the criteria, we'll accept you for screening. And when you start the screening process, the very first thing we have to do is consent you to the trial. Consent doesn't mean that you're definitely going to get the therapy. It just means that we can evaluate your child to see whether it would be safe to give you the therapy. So that's, and you know, it's a really difficult period that. I wouldn't ever invite somebody to screen if I didn't think there was a good chance that they were going to be eligible, but there is the risk that something will come up that will mean that they're not eligible or it wouldn't be safe to proceed. And the screening process is all about making sure that your child meets the eligibility criteria. And it really is about safety. It's if I'm going to put a needle into your head, you know, is that a safe thing? Is it as safe as it possibly can be for your child? So it may include a scan where we just look at the physical structure of the brain. You know, is it going to be practical that I can get a needle into that space or not? Because I'm not going to put them through further invasive procedures to then discover that I'm actually causing harm. So it really is about safety. The inclusion and exclusion criteria, and I know that there have been a few questions about why so few patients get into clinical trials, why at the beginning are there, you know, such strict inclusion and exclusion criteria. And my boss would always taught me, clinical trials are really selfish. They're not, in the long run, they're about the condition and about improving therapy for patients. But in the short term, they're answering a question. And in phase 1-2 trials, this is safety. is this a safe therapy delivered in a safe way? And later on we'll ask the question of whether it helps or not. So we're trying to ask that question. So that's why it's quite rigid. Can it be tolerated? So it's safety and tolerability. So there's side effects with everything. We're going to give an immunosuppression regime, which is not ideal. But is that okay within the grand scheme of things is really what we're saying. We're also thinking about dose, and so in these early phase trials we might have a low dose, we might have a high dose, we may have 3 dose arms, and so we're trying to decide what's the safest tolerable dose of that therapy. And then later we're going to think about benefit. We're going to capture as much efficacy data and benefit data as we can, but really at this point in time it's all about safety. And we need all of our patients to be as similar to one another as we possibly can make them, so that we can explain any differences as a result of the therapy and the intervention, rather than as a result of the differences between those participants. So a lot of the time, and this is one of the worst parts of my job, is that I'm telling families, actually, this isn't appropriate for your child. And it's one of the worst parts, you know, and it's worse in some of our conditions where I'm saying, I have nothing and I'm not even going to give them a chance. And that's what it feels like to parents. And it feels like looking at that mountain. It's really awful. And I know that it's worse for you guys. I think you guys are in a better position because there's more coming for your children and they've got time. So when we are evaluating them, we need to measure lots and lots of things, not just for safety, but also to judge the efficacy, so how well this works and how much benefit we can get. So that'll involve blood tests, urine tests, CSF tests, so lots of investigations. It'll involve physiotherapy assessments, cognitive tests, questionnaires. Families get sick of being given so many questionnaires, but we have to do it. And sometimes videos. It's lots of hospital visits. It's tests that your kid wouldn't otherwise have. Some of the tests have risks. You know, if I'm putting a needle in their back, that's a risk. There's an invasive procedure. There's some things that your child just won't want to do. And we can manoeuvre as much as we can. We're paediatricians, so we're used to that. Sometimes we get results that we don't expect, and sometimes we get results we don't know what to do with. Sometimes we get a result which means I have to then repeat a test. And I remember I had a family that had travelled, so we do lots of international trials. I had a family that had come from Malta, and we'd done a test which was a safety test, and the results were a bit inconclusive. And the clinical trial sponsor, so the company, phoned me at 3:00 on a Friday afternoon and said, you need to repeat it, while they were on the way to the airport to go home. And I had to cancel their flights and say, no, come back to the hospital because we need to repeat this. That's horrible for a family to go through for something that we can't really explain. And often there are results that you're never going to see in a trial. Results that I'm never going to see. So half the blood tests that we do, the urine tests we do, they get sent off to a central lab somewhere in the world for the company that are running the trial to analyse for the benefit of the clinical trial development programme, but they don't send me those results. And they're not results I can share with you because I don't have them either. And that's really frustrating for families because you want to know what's happening. You want to know, is this gene therapy working or not? And all I can say to you is, well, I think, yeah, maybe, or I'm not so sure. And that's hard. So some of the other experiences that have shaped how I deliver clinical trials, because like I say, every time I do one, I change my approach to the next patient. I have to, because I have to learn from every patient I meet and every experience I have. So I've had children that have reacted to the immunosuppression. I had one little girl who was on a really standard immunosuppression regime. She got an esophageal kind of inflammatory response. She was being sick, she had a rash. She was really miserable. Her mouth was full of ulcers. Really horrible experience. I've had a child that we brought from another country who deteriorated really significantly while we were screening. We hadn't even got as far as dosing, and that was awful. We've had a couple of siblings who we gave a compassionate access gene therapy to, and to be honest, the gene therapy product was just rubbish. That's why I keep always mentioning it needs to be good. It can't just be any product, it has to be a good working product. This one, it's done something, but it's not good enough. And the concept behind it was really good, but the actual product was crap. We've had this multiple times, not necessarily with gene therapy trials, but with other trials, where the trial overall has failed to meet what we call the primary outcomes. So this is what the regulators are going to judge your trial on, your therapy on. So it's not met the primary outcomes, but we've thought actually if you designed the trial a bit differently, you'd done X, Y, and Z a bit differently, we actually think that therapy would have worked. And so now that company is not going to pursue that therapy because it's not in their financial interests, because it was badly designed in the first place. Other immune responses to therapy, so we've seen liver enzyme tests rise in one of our patients, which has meant that we don't really know what it means, but we worry that it's a rejection to the gene therapy. So I've put them on an extended course of immunosuppression in an attempt to salvage a response. We've had one company went into bankruptcy essentially, had to withdraw, and so we were left with a group of children that we'd given gene therapy to but had no follow-up in place. And so that's been really miserable. And similarly, a trial again badly designed, so we had a group of children who all were very different, and so the company kind of felt that they could no longer continue to recruit because they couldn't tell whether they were doing good or not. We've had children who have had their underlying disease deteriorate during follow-up, and again we've had to try and repatriate them back to the countries that they've come from. And commercial sponsors withdrawing because they've not seen benefit quickly enough. So this yellow box should say something about other questions I've been asked when I've been doing trials, which might just be helpful for you guys. Often I'll get an email from somebody very wealthy somewhere in the world saying, can I just pay? I'll crowdfund or I'll pay. I've got a really wealthy uncle who will pay for this gene therapy, and there is no mechanism by which I can offer you a gene therapy if you pay me for it. It's just not an option. So don't crowdfund for gene therapy. People that, children enrolled, and they'll say, do they really have to have this test? Usually the answer is yes, they do. It's just, we try and be as flexible, we try and be as creative about how we do things to minimize the burden to you and to your child, but we have to be a bit rigid. Clinical trials have to be rigid because we have to see uniform data, so we have to be quite scientific about it. Can we come for the study visit on a different day? Usually the answer is yes. We'll try and accommodate anything. We'll try and be really supportive and make this work within the family, but it has to be a two-way street. So if you're participating in a clinical trial, we need you to give as much as you possibly can, and we'll try and support you with it. Often families come and say, well, they had a scan a year or so ago, or we've got this test already, do we really need to repeat it? Unfortunately, we usually do have to repeat it because all of our scanners, all of our tests have to go through a kind of approval process so that all of the scans that are compared from wherever you are in the world are comparable. So the same quality, the same sequences. And so unfortunately, we do have to repeat a lot of tests that you may have already had done. Do I need to move house to participate? Usually the answer is no. In the US, often they do expect people to relocate to a centre in the US if it's an international trial. For us, we would just expect that usually for gene therapy trials, we have families that stay with us for at least a month, usually 2 months, around the period of dosing. And so that, in the UK, that might be a bit more flexible, and it might be that we say that you have to be within 2 hours, and we always make sure that that is supported. So I'll never take on a clinical trial with a company unless they guarantee me that they're going to fund travel, accommodation expenses, etc. for families. And I would really encourage you to, if anybody tells you you're not entitled to that, you need to raise that with the lead at whatever site you're at. And that comes to is there any financial help to attend study visits? Absolutely, we should not be delivering clinical trials and not supporting you in doing so. So there should always be funding available to you for accommodation, travel, food when you're there, family members to be present with you. And if anybody does participate in a clinical trial, never go as a solo parent. Take somebody. Take your best friend, take a grandparent, anybody that's going to support you, because you need support as a parent there dealing with this. So those would be my take-home comments. So things I want you to know, and don't be too depressed. Sometimes we do these and we cause harm. That's being really honest. And so it's not, I just don't want you to take this lightly. It's exciting, the prospect of gene therapy, a therapy that's going to change your child's experience. But sometimes in doing this, we cause harm, and we have to be honest about that. And experimental therapy doesn't always mean a better therapy. Just because it's new and it's exciting and it says gene therapy doesn't mean that it's necessarily going to be better than the supportive therapy your child's receiving right now. And there might be changes during a trial. We can't predict those. So I don't know when the protocol is going to change. I don't know what's going to happen with patients recruited in another country that may adapt the protocol, which may be for the better, but it may mean that it becomes more intensive as we go through. But we ask you to stick with us if you're participating. And this is another really important point, which I don't think we would have the same problem in the UK, but we're delivering these clinical trials still within the NHS, and you all know what the NHS is like. We try really hard, but it is a very much overburdened, stretched service, and we're using our experts from within the NHS to be able to deliver these therapies. Sometimes, particularly my experience with international participants, is that they think it's a private service being part of a clinical trial, and it is a much better service in some ways, and it's more intensive, and we put you on lovely trials units, which are fabulous places to be, but it's still the NHS. So you still have to bear with us when things are slow or things don't move as quickly as you would ideally like. And I have that battle myself, that this is not what I imagine clinical trials to be like. So I know that doesn't feel bright and smiley. I do believe in gene therapy, and I wouldn't be doing the trials if I didn't. But we just have to be really honest about it, and we have to be realistic about it. And if we do it badly, if we don't do research well, we're going to fail kids in the future. And if we don't do the research, we're never going to have a therapy. So we do have to keep going with all of this, and I genuinely believe we can, but I just want us to be mindful of the challenges and the burdens that comes with. So this is the team that I work with in Manchester. So we have a clinical trials kind of neurogenetics team. Some of you know Sid Banker, who's our geneticist and runs our Rett's clinic. And these are other members of our team from neurology and genetics. And Simon and I work in these rare neurodegenerative clinical trials. So I will happily take any questions if you want.